I have the pleasure of introducing you to Deborah Hartman. She is the Global Scientific Head of the Orexin Program at Centessa Pharmaceuticals. Deborah is responsible for helping to shape the overall research strategy to investigate orexin agonists as potential new treatments. She received her bachelor's degree at Princeton University and a PhD from Yale University in the area of molecular and cellular biology. For over 20 years, Deborah has been involved in research targeting neurological and psychiatric disorders. Prior to joining Centessa, Deborah held senior leadership roles in other biotech and pharmaceutical companies including Takeda, AstraZeneca, and Hoffmann Roche. She is now focused on developing new potential treatments for narcolepsy and idiopathic hypersomnia, which she'll tell us about today. Debbie, it is so good to see you. I enjoy every moment that I get to spend with you over the last few years, and I know that you've been a very welcome presenter at our conferences with the Hypersomnia Foundation and spoken to many people in our community, and I just want to welcome you to this platform, and we're so excited to hear from you again and learn all the new things that you're ready to share with our community. So thank you for taking the time to spend with us today.
Thank you, Claire. Thank you so much for the invitation to be part of this Living With podcast series. It's so great to see you, and I'm so impressed to see everything that you and the Hypersomnia Foundation are doing to promote research and development of new medicines and to support the community. As a scientist, I believe there is such good reason for hope now that new discoveries may come to help to transform treatment of sleep-wake disorders. And so just happy to be here to talk about that.
Yeah, thank you. So to give our audience some background about you and what you do and what you specialize in, can you tell us why you decided to become a drug discovery scientist? And in particular, what led you to Orexin?
Yeah, I guess as a student, I was always, since I was pretty young, fascinated by how the brain works. I ended up doing my PhD in molecular neurobiology, and then kind of a set of circumstances led me to decide to pursue a career in drug discovery, where I discovered a lot of just cross-disciplinary interaction, working with so many different scientists, different disciplines, and really realized that, I had this passion to try to develop new medicines. Recently, as you know, I've been really excited by the potential for orexin agonists in particular for the treatment of narcolepsy, for treatment of IH and other disorders as well. And that was just a research project that I was asked to lead in a former company. And as the results started to come in and we started to learn more about orexin physiology and about orexin agonists, that's just been my passion now for about 8 years.
But you've been working in sleep research for much longer. How long?
It's probably almost 10 years now. And, recently I've really, before that, I worked in central nervous system disorders more broadly. But when I discovered the world of orexin agonists, that's when I really started to learn a lot more detail about sleep disorders, specifically narcolepsy and idiopathic hypersomnia. And at the moment, I'm the scientific lead for an orexin agonist program at Centessa Pharmaceuticals. So I'm a scientist doing drug discovery research, and in that program, we've now moved our first orexin agonist molecule into phase 1 clinical trials. So I work in the research space as well as the early clinical development space.
Yeah, it's really interesting actually that you mentioned both narcolepsy and IH, because I think traditionally there's been sort of more of a kind of focus on narcolepsy, and IH has sort of been a little bit in the sidelines. So can you speak to how the orexin agonists and the drug development process is relevant to the IH community?
Sure. So I think when we think about orexin agonists, both narcolepsy and IH are really particularly relevant. As a scientist focused on drug discovery, for me, I recognize the importance in IH of having this tremendous need in the community for better options, and also this lack of understanding about what causes IH. And the Hypersomnia Foundation yourself, Claire, I've heard you many times challenge the entire research community to take idiopathic out of idiopathic hypersomnia, which is a really powerful concept. Now, I guess to explain more what that means for people in general, the word idiopathic, of course, means that IH is a disorder without a known cause. And in fact, IH may represent people with similar symptoms, but actually those symptoms might be caused by different things. So recent data is starting to suggest that orexin agonists might be helpful in treating IH. And this is very exciting, first and foremost, for those living with IH, because it could mean a potential new treatment option where there hasn't been one until now. And for me, on a personal level, it's really thrilling to work on this entirely new class of medicines, and to see as well that as we learn more about IH, that the prevalence may actually be higher than previously thought. So perhaps even more people are suffering from IH than previously thought, which means that the unmet need is really great. So this novel
really
science,
huge.
overall, very exciting for patients, for the medical community, and for those of us doing research.
Yeah. Thank you for bringing that up, because I think, over the last year, we've been also really challenged by recent research, Dr. Plant, with the incidence of IH being significantly higher than we previously thought. And for anyone listening here, you can actually find that presentation by Dr. Plant on our website and YouTube, because it really is a very significant shift in perceptions of incidence. But how do you kind of differentiate, Deborah, narcolepsy from IH? What would be helpful as an explanation to anyone listening to this if they weren't quite sure? Because sometimes the lines are really blurred and it's really confusing.
Yeah, no, that's for sure. So I'll explain it to the extent that I understand it. And, starting with the fact that both IH and narcolepsy are characterized by excessive daytime sleepiness as one of the absolute overriding symptoms. If we start with idiopathic hypersomnia, individuals with IH may need 11 hours or more sleep per night, to my understanding, and might find it extremely difficult to wake up, which is a symptom that we often call sleep inertia. In addition, in IH, there may be an uncontrollable desire to go back to sleep, and many people feel confusion and irritability when they are sleeping, which sometimes is called sleep drunkenness. Symptoms are different, but somewhat overlapping between IH and narcolepsy, where in narcolepsy type 1, individuals experience excessive sleepiness during the day, but they often have disturbed sleep at night. In NT1, individuals also experience cataplexy, which is this sudden and transient loss of muscle tone that can occur in arm, leg, facial muscles, and can also result in a full body collapse. Narcolepsy type 1 is known to be caused by the loss of orexin in the brain, and this has really opened up a whole new avenue of research towards developing new medicines, which is what we're talking about today. In people with narcolepsy type 2, what we find is that some individuals have symptoms more similar to IH, and others have symptoms more similar to NT1. But individuals with narcolepsy do not experience cataplexy. That's quite unique to NT1. If you think about narcolepsy type 2 in terms of orexin levels in the brain can be completely normal in NT2, or in some cases be only partially reduced. And just like IH, the cause of narcolepsy type 2 is still unknown. One last point I guess that I would make is that in both IH and narcolepsy, individuals often describe problems with memory or problems with an ability to pay attention or concentrate, and this is quite broadly referred to as brain fog. Now, brain fog might be somewhat different between IH and narcolepsy, and it's really not well defined in either case. Much more research is needed to understand how to address this and really how important this is in impacting the lives of people living with both IH and narcolepsy.
Yeah, it's really interesting. So I think, traditionally when we think about the research and the patient experience in the doctor's office, so we always check off all the main symptoms. and then kind of come out thinking, okay, well, maybe that's kind of sufficient. But actually the patient experience and the patient voice, that is so much coming to the foreground and kind of communicating that actually there's so much more than like what's been traditionally thought as challenging in terms of symptoms. So brain fog is something that we hear about over and over and over again, and kind of also alludes to like the massive impact of living with a sleep disorder on every aspect of one's life. So we're always grateful for scientists like you, Deborah, who kind of recognize that on a fundamental level. This isn't just about like checking off, the symptomology and, okay, we're done, here's the medication, off you go. And it's like understanding the patient experience to be so much more kind of prevalent throughout their entire life. So with that, what would you say, like the areas of research interest in IH? What's the direction? Where is it at? And what's the direction it's going in for our idiopathic hypersomnia community?
I guess for me, there are three key questions that scientists and physicians are exploring today regarding idiopathic hypersomnia. The first is, what is the underlying cause of IH? Secondly, could there be different causes that result in the pattern of symptoms that are diagnosed today as IH? So maybe not a single cause, but maybe multiple different causes. And then thirdly, how can we explain the diversity of symptoms and severity that are experienced by different people with IH? And as we understand sleep physiology, and we learn more about sleep physiology from even our experiences now with orexin and orexin agonists, there's a couple different theories or hypotheses related to IH that scientists and researchers are trying to test. On the one hand, it might be that those pathways in the brain that promote sleep, perhaps they're overactive in IH and really drive and increase this daytime sleepiness. On the other hand, perhaps the signaling pathways that normally keep us awake during the day are not active enough in IH. And therefore, we're sleepy because we just can't get that wake signal sufficiently in the brain. So there's many different ways to approach this. And I wanted to share two examples of some research that has been done that are looking at IH and are related to orexin biology. The first is an example of an animal model that was developed in the research group of Professor Yanagisawa, who created a mouse model that they call sleepy mice. So sleepy mice have been genetically modified to have a splicing mutation in a gene called SIK3. And this leads to increased sleep time and an increased need for sleep in these mice. They can only stay awake for a few hours a day. The researchers reported that in these mice with this genetic mutation that they are using as a model for IH, they took an orexin agonist and treated those mice, and they found that the orexin agonist worked. In terms of decreasing sleepiness, the mice were able to stay awake with more normal periods of wakefulness. So this showed that an orexin agonist, at least in this genetic model, was able to still have effects in terms of increasing wakefulness, even though the orexin levels, for instance, were not changed. Another example is from human genetic studies. So in the group of Professor Makoto Handa, they described a very rare genetic mutation that was associated with IH. And this rare mutation, found in a very small number of individuals with IH, actually resulted in an unusual form of the orexin peptide itself. So this particular version of the orexin peptide in these individuals was weaker or less efficient in activating orexin receptors. That suggests that in IH, there may, in some cases, be a reduced efficiency of orexin signaling in the brain. And if that's the case, then again, perhaps an orexin agonist might reduce symptoms by restoring or then normalizing orexin signaling in the brain by further enhancing and increasing that signal, which is reduced or weakened in those individuals. So much more research is needed, but importantly, these results do provide hypotheses that can be tested with orexin agonists in clinical trials in IH.
It's really interesting that the focus of research is so pointed in that direction. But is there any kind of clinical evidence, Deborah, to support those hypotheses?
Yes, orexin agonists have now been tested in a few studies with small numbers of individuals with IH. The very first clinical study of an orexin agonist was actually in IH, was actually just published last year. The orexin agonist that was tested was called danephoretin, and the lead author on the paper was Professor Emmanuel Mignot from Stanford, who I know many people are familiar with. Dr. Mignot discovered the link between orexin and NT1 over 20 years ago. So in this study, which was a relatively small study enrolling about 28 participants with IH, they used a crossover design so that each person received a single dose of the orexin agonist, and then on a different day, maybe 2 days later, a single dose of placebo, and that was randomized. So the individual didn't know whether they received the placebo first or the orexin agonist first. And then the researchers could compare the wakefulness that was produced between the orexin agonist and the placebo treatment. So, big question was, what didn't the orexin agonist actually reduce sleepiness or enhance wakefulness in IH? And the answer, as you can see in this paper, was a resounding yes. In fact, both wakefulness and attention levels were increased in the individuals with IH after treatment with the orexin agonist compared to placebo. And about 40% of the participants in the study also reported mild or moderate adverse events, the most common being urinary urgency. And beyond that, the orexin agonist was fairly well tolerated. So from this study, we learned that orexin agonists may be very effective in reducing symptoms in IH, despite the fact that orexin levels are normal. And this is consistent with the hypothesis proposed by the human genetic study, as well as that mouse model, which suggested that in IH, perhaps in some cases, this may be associated not with a loss of orexin, but with normal orexin levels and perhaps weakened orexin signaling.
Yeah, it's so interesting. It sort of doesn't make sense at face value, does it? But with that, can you explain how the orexin agonist works and what its role is in maintaining wakefulness?
Orexin is a neuropeptide in the brain. There are two orexin molecules, orexin A and orexin B, and orexin serves as a powerful signaling molecule in the brain, which acts as what you could think of as the conductor of the orchestra for wakefulness. Orexin will strongly suppress both REM, rapid eye movement sleep, and non-REM sleep during the day, and it coordinates the activity of multiple neurotransmitter pathways in the brain, which together promote wakefulness. So you can imagine orexin keeping all the different sections of the orchestra, the wakefulness orchestra, active and playing in harmony so that we can stay awake and alert during the day. The neurotransmitters that are involved downstream of orexin, the players in the orchestra or instruments in the orchestra, are probably familiar to many people. They include neurotransmitters such as norepinephrine, histamine, dopamine, acetylcholine, and serotonin. So if you think about stimulants and other wake-promoting medications, these act somewhat differently in that they activate only one or a few of these downstream neurotransmitter pathways, but they lack the ability to coordinate wakefulness across the entire orchestra of neurotransmitters the way that orexin is able to do. So the idea is that with orexin agonists, we hope to develop more effective medicines by harnessing all the wake-promoting activity in the brain and reduce excessive daytime sleepiness in the most effective way possible. Now, it's an exciting area, and there certainly are multiple different companies now are advancing orexin agonists into clinical studies. So there's hope that, in the future, some of this research may be translated into new medicines. Certainly, Centessa, where I work, has an orexin agonist program, and the orexin agonists in development right now are all selectively activating the orexin 2 receptor, which is one of two receptors in the brain. There's an orexin 1 and orexin 2 receptor. And this orexin 2 receptor is the receptor that is most strongly associated then with wakefulness.
That's so interesting. So I have another question for you related to this. How do you see going forward in terms of the science and the role, kind of interacting the science with the patient experience? Because as we said earlier, that there is a shift, isn't there, in sort of culture around incorporating the patient the lived experience of everybody into, kind of meaningful research. How do you see that role kind of developing?
Yeah, I think this is a really important area, and really happy to share my view, but also very interested in yours, Claire. Maybe after I share mine, you can make a few comments from your perspective as well, definitely. And how important this is.
Sure.
So I think in the near future, we're really hopeful for new and better treatments based on orexin agonist molecules, and that this, together with better understanding of the underlying causes of IH and better tools for diagnosis, will all come together. But as a drug developer and a researcher for quite a few years now, I really have come to realize that it is the people who are living with IH, narcolepsy, and other sleep-wake disorders, as well as almost any other indication, who play a really critical role in drug development. We cannot do in industry or in research, we cannot fully investigate or develop new medicines without having that input. And when I look to the future, I see a really important trend, as you've just mentioned, Claire, towards integrating that patient experience directly into drug development programs. So for me, for instance, at Centessa, we've included and tried to encourage advocate voices into our research programs, even at the earliest stages of development. But as an industry overall, this is just one piece of the puzzle. We need to listen even more carefully and learn more deeply about that lived experience. And this is critical when it comes to the design of clinical trials or the selection of clinical outcome measures. What are we measuring? And does it really matter what the result is of that study to how that might impact the individual's life when they're taking that medicine?
Only the patient can really answer that question. What's so important? When you talk about outcomes, you could imagine a scenario where a company's just like pitching in the dark, like these are the clinical outcomes we imagine, but actually if you understand the patient experience, that might be, considerably different.
Absolutely. And it really needs that partnership, that collaboration with individuals who are living with that every day, who can help us to understand the impact, what's really important, and ultimately, together then create new medicines that really matter and drugs that will make a meaningful difference, in the lives of those who are waiting for them and who really need them.
Yeah, it's, I've certainly seen that shift. I think it's, I would say cast our minds back. I think probably you and I met maybe as far back as 2018 or something. And even since then, there's been such kind of cultural and tangible shift in engaging patients at drug development level, clinical trials. And actually just, if anyone is listening to this and wants to sort of understand a bit more what's available in terms of clinical trials, it is on the Hypersomnia Foundation website. So there are the trials that are listed there are open, and you can engage with those and have a look and see whether you may qualify. And just to kind of plug, I suppose, that the role of engaging in clinical trials is so important to advance research, and that's why we need the partnership, right, with companies like Centessa. with patient advocacy groups like the Hypersomnia Foundation kind of right in the middle there, intersecting between our partners, but also obviously advocating fully for the patients and caregivers and loved ones and supporters. So there's a lot of work to be done, but I do kind of feel encouraged actually sort of personally and professionally that there is that shift towards listening and incorporating the patient experience into trials with a view to actually having a medication that, as you said, hits a lot of the, of course, core symptoms, but the massive impact that living with a sleep disorder has on multiple areas of any given 24 hours, whether it's, of course, sleep, waking, quality of wakefulness, relationships, jobs, all the things that are so important and significant and so severely impacted and so invisible. So Deborah, I've always had an incredible amount of respect for what not just what you do as a brilliant scientist, but actually your posture and ear towards the patient experience. And I do remember the first time I met you thinking, I don't think a scientist or a doctor's ever sat down and had an hour and a half and just listening to what it's like to live with then a child with type 1 narcolepsy, now sort of an adult. But so thank you from sort of on behalf of the community, really, because I think you've also been important in shifting some of that culture, like the traditional scientist, you know, in the lab, in the office, to actually really, really genuinely engaging with the patient community and with a real heartfelt kind of posture towards basically what we understand as suffering, actually. So I'd like to thank you for that, Deborah, and for your expertise, of course, which always explain in such tangible ways. Thank goodness for that, because it is complex, isn't it? And it's really difficult sometimes to now differentiate between, you know, whether it's the Orexin 1 or 2 and IH and NT2. It's actually sometimes sounds more confusing right now than it did a few years ago. But thank you for shedding light on those differences for us today.
Thank you, Claire, and thank you to you and to really the entire community who has also embraced this. There's a courage to be so open and transparent as well for people to share their stories, and that is absolutely, it's essential, but it is so well appreciated, and it motivates all of us doing research in an incredible way. Now we know, you know, when we wake up in the morning, what we're aiming to do and why. And sometimes you have a picture of somebody in your head, just knowing, you know, I'm going to try to do my best today to try to make this, you know, a step forward today, this week, this year, etc. So I really like to thank everybody who has been so willing to share their stories. And there's no way that we will know whether anything that we develop works unless we can get that feedback from a clinical trial, as you say. So that's really, really important that we design them well, that we're measuring the right things, and that we make them accessible to people to participate.
Yeah. And it is really exciting for the idiopathic hypersomnia community, because I think historically you know, sometimes, as I said earlier, you know, they're a little bit dragging behind, but I'm really encouraged by how IH is front and center right now with the Orexin Agonists, with, you know, even without a full understanding of, you know, as you said earlier, let's take the idiopathic out of idiopathic hypersomnia. But it's really encouraging to think that they're in the, you know, they're front runners as well for the new medications that are coming out, and not just sitting in the back rows waiting for it to work in narcolepsy. So hopeful for both conditions and both communities to be significantly helped with this medication. So thank you for all you do in the lab and in the office. Okay.